video I am going to discuss about the glycogen degradation. In the last video we saw the glycogen synthesis, that is glycogenolysis, where glucose 1-phosphate was required to prepare UTP glucose, from where primer will be synthesized, and on that primer glycogen synthesis will act and elongate the chain. But glycogen is a branched chain polysaccharide. So branching must be there. So for the formation of branches, a separate set of branching enzyme is required, and that is 4,6 transferase. So in glycogen synthesis, the first thing that we required was glucose 1-phosphate, and this was prepared from glucose to glucose 6-phosphate. This glucose 6-phosphate was isomerized to glucose 1-phosphate. Next we required UTP. These two form UTP glucose. This UTP, that is uridine triphosphate and glucose 1-phosphate, combined together forming UDP glucose. Now we require a primer because glycogen synthesis cannot initiate the chain formation. So another enzyme called glycogen, this glycogen will accept this glucose moiety from UTP glucose, and it will form a 4-molecule chain. And this is where the enzyme glycogen synthase will act. On this small primer, this glycogen synthase will act, and it will form 1,4 alpha 1,4 linkage. So it will form alpha 1,4 linkage and it will elongate the chain. But glycogen is a branched chain polysaccharide. So branches must be formed, and that is done by the branching enzyme, that is 4:6 transferase. That is amylo alpha 1,6 amylo alpha 1,4 alpha 1,6 transglucosylase. Now the function of this enzyme was to break 6 to 8 units of glucose from the chain, and it will then attach it with the non-terminal carbon of the chain, and it will form 1,4 linkages. Thus the branching will be formed, and that elongation of that branched part will be done by glycogen synthesis, because that is responsible for the formation of this alpha 1,6 bond. Now in case of glycogenolysis, that means glycogen degradation, the product that will be formed will be glucose 1-phosphate. That's obvious, because glucose 1-phosphate, from glucose 1-phosphate we were getting glycogen. So the first product that will be formed, or the end product we can say, that is glucose 1-phosphate. At the same time glucose will also be produced, free glucose without any phosphate group attached. So the enzyme that is required for the breakdown of this glycogen chain is glycogen phosphorylase. Glycogen phosphorylase. So this is the enzyme that was breaking the alpha 1,4 linkages, like that of glycogen synthase, which was making the alpha 1,4 linkages. So it is cleaves alpha 1,4 linkage. Same for the branching part. Here we require a different set of enzyme for the formation of branch. The branches must also be removed from the glycogen. So for that, separate set of debranching enzymes are also present. So debranching branching enzyme which will remove the branches. So it helps in the removal of branches, because this can cleave alpha 1,4 linkages, but it cannot remove the branches. So for that, first enzyme that is required, oligo alpha 1,4 alpha 1,4 glucan transferase. What is happening is that, suppose these are the glucose units that are present in the chain. Now from here, these are the branches. These are the branches. Now the bond between this glucose and this glucose, in between them they have alpha 1,6 linkages. They have alpha 1,6 linkages, and in between these, like in the chain of the branched, this branched part, in between them they have alpha 1,4 linkages that is extended by glycogen synthase. Now this branches needs to be removed. For that, we have to transfer these branches, these to the chain, to the main chain, so that all the branches are removed and a long chain is formed ultimately. So what is happening? Suppose there are 4 residues of glucose. I have drawn 4 units. So out of these 4, 3 will be removed. These 3 will be removed. So the breaking of these alpha 1,4 linkages will take place. So that's why alpha 1,4. Then it will come here and it will again form alpha 1,4 linkages. So again alpha 1,4. So that's why this is also called 4:4 transferase. 4:4 transferase. Same here. There are 5 units, so these 4 will be going to the go and join to the main chain. So these 4 units, the bond between this is the alpha 1,4 bond. So this will break and this will go and join the main chain, forming again alpha 1,4 linkages. So ultimately a long chain is formed on which this enzyme glycogen phosphorylase will act and cleave the alpha 1,4 bond. So now these are gone. So I am drawing the 3 glucose were here. So I am drawing 3 here, 4. So these 3 they have come and joined the main chain. Now what about the remaining glucose? This glucose that is left behind, this one and this one, that is removed by another set of enzyme, and that is amylo alpha 1,6 glycosidase. This is an acid maltase enzyme. This will break the 1,6 linkages. So as I have told you that this bond is formed via 1,6 bond, and the remaining these are 1,4 linkages. So this 1,6 linkage is broken down by this enzyme. So this enzyme will cleave this bond and remove this glucose moiety as free glucose. So this glucose moiety will be removed as free glucose. No phosphate group will be attached. But when glycogen phosphorylase will act on this chain, this will give glucose 1-phosphate. When 1,4 linkages will be broken, each and every unit will give rise to glucose 1-phosphate. But this one the last remaining glucose moiety that is attached to the main chain, their bond will be cleaved and it will give rise to free glucose. So this is your debranching enzyme. So this is how, so these two are removed now. So you can see the resultant long chain is formed on which glycogen phosphorylase will act, cleave the 1,4 linkages that are present in these long chain and liberate glucose 1-phosphate. Now another set of enzyme apart from this, that is lysosomal enzyme, which also helps in the degradation of glycogen. Lysosomal enzyme degrades about 1 to 3% of glycogen. So 1 to 3% glycogen is degraded by lysosomal enzyme. It is degraded by lysosomal enzyme, and the name of this enzyme is acid alpha-1,4 glucosidase. So this enzyme breaks down glycogen into glucose molecules. So this is your different enzymes which act on the glycogen and that produces glucose 1-phosphate. Now the next step is the where glucose 1-phosphate is converted to glucose 6-phosphate. So next one is isomerization of glucose 1-phosphate to glucose 6-phosphate. As I have said that glucose 6-phosphate is isomerized to glucose 1-phosphate. Same way, so this glucose 1-phosphate converted to glucose 6-phosphate by phosphoglucomutase enzyme. And this glucose 6-phosphate is acted by the enzyme glucose 6-phosphatase producing glucose. So this glycogenolysis is occurring when the blood glucose level is low, when the cell's energy need is high. So glucose will be produced. So in the fasting stage majorly this glycogenolysis takes place. We saw that glycogen is first broken down by the enzyme glycogen phosphorylase, which produces glucose 1-phosphate, and for the removal of branches, two enzymes, that is oligo alpha-1,4 alpha-1,4 glucan transferase, or we can say 4:4. Why 4:4? Because alpha-1,4 linkages is broken in the branching portion, then that is going and joining with the main chain, that is again forming alpha-1,4 linkages. That's why 4:4 transferase. On the other hand, the last glucose moiety that is attached to the main chain via the 1,6 linkages, that is removed by another set of enzyme, that is amino alpha-1,6 glucosidase, and this gives rise to free glucose, no phosphorylated form. And glycogen phosphorylase act on this long chain now, producing glucose 1-phosphate, which is isomerized to glucose 6-phosphate. So this is all about your glycogenolysis. In the next video, I will discuss about the regulation of these two processes. Thank you.